for your lab practical 1, you do need to know your mitosis phases, know your mitosis phases in order, know how to identify some of the features within each phase, and also know what is happening in each phase. Okay, so this first one here, of course, is your interphase. Interphase is your initial cell growth. Of course, you have duplication of the organelles that's happening, replication of your DNA, and continuous cell growth. Okay, so you need to know this is your plasma membrane, this is your nucleus, nucleolus, and these two dots are your centrioles. Okay, next we have prophase, and we have with the models early and late prophase. Early prophase is the first one here. In early prophase, what is happening is the nuclear membrane here starts to degenerate and disappear. The chromatin starts to condense and form chromosomes, okay, which are the gray lines here, are the chromosomes. The two black dots again are your centrioles. The two centrioles start to move apart, and it starts to form your mitotic spindle, or your spindle fibers. Okay, in your late prophase, if you notice here, the nuclear membrane has degenerated and disappeared. Your centrioles have moved to opposite poles of the cell. Your metaphase, what happens is the chromosomes, of course, line up in the middle of the cell on the metaphase plate. Okay, so to help you to remember metaphase, just remember that metaphase starts with an M. They're lining up in the middle, starts with an M. On the metaphase plate, starts with an M. Okay. Then, of course, you have your centrioles here. Okay, they're still on opposite poles of the cell. Okay, next you have your anaphase. On the models for anaphase, you do have early and late. But during anaphase, what you see is the chromosomes start to move towards opposite poles of the cell as the spindle fibers shorten. So they're going to be pulling apart. So the chromosomes pull apart. And you see, of course, the mitotic spindle, of course, in the middle has shortened. Centrioles are still on opposite poles of the cell. Okay, in late anaphase, of course, noticeable that the chromatids, of course, are more V-shaped because they're moving further apart. Okay, this is your mitotic spindle, centrioles, and right here, this little indentation, of course, is your cleavage furrow. Okay, in telophase, of course, you have, of course, the cleavage furrow is this little indentation here of the cell, and the cleavage furrow, of course, is where the cell is pinching into to eventually become two identical daughter cells at the end of telophase. Okay, so if you notice here, you have your centrioles on each pole of the cell. The mitotic spindle or spindle fibers lighten, okay, and become less visible. Also, during this stage, the nuclear membranes on each side here begin to reassemble. Again, the mitotic spindle starts to become less visible, and eventually your cytokinesis is going to be completed. So what happens is the cleavage furrow will pinch the cell in two, and then it will become two daughter cells. Okay, so this model is showing you that cytokinesis has completed, and you have two new daughter cells. Here's one cell, here's the second cell. Okay. When it is has completed into two new cells, again, they become two daughter cells interphase. Okay, so they're doing interphase all over again.